Today we're going to be looking at common food items and testing them for the biological molecules that we've been discussing in class. We're also going to dig in a little bit more to some of the terms that are commonly used in science, specifically the terms positive control and negative control. As you might recall, the four classes of biological molecules are carbohydrates, proteins, lipids, and nucleic acids. We're going to test for both simple sugars, which are monosaccharides and disaccharides, and starches, which is a polysaccharide. That's for our carbohydrates. We're also going to be testing for proteins and lipids. However, we are not going to test for nucleic acids. Can you figure out why? Now, I'm going to set up the lab for you. The indicator solutions that we're going to need for this lab are carcinogenic, so that's why I'm doing it here when I have gloves and goggles and all the safety equipment around me. But you're going to have access to all the data, and so you can do the same analysis that you would if you were actually in the classroom physically. So our first indicator solution that we're going to use is activated only when it's heated. So the first thing I'm going to do is actually get that set up. I've got my beaker. I'm going to fill it up about halfway with regular tap water and get it cooking on this hot plate. Okay. And I'm just going to turn that all the way up. Once it starts to boil, then I can turn it down just like you might do if you were cooking pasta. I'm going to add some of these boiling chips. This is a lab safety consideration to prevent this beaker from boiling over. So they only need a couple of three. What is a couple of three? First thing that I need to do is draw a line about 1 centimeter up from the bottom of the tube. This part takes forever, but luckily you get to have time lapse. Now that I've got all the lines drawn along the bottoms of these test tubes, I can do the most important part of science, which is labeling things. So I have 4 foods that I'm going to test. So the first thing that I'm going to do is label all of the tubes with a particular food that is correct for them. All right, another time lapse for you, lucky you. Pro tip, if you ever need a Sharpie eraser, alcohol swabs. I'm going to walk you through setting up the lettuce test. And then I'm just going to repeat that process for all the other foods instead of making you sit here and watch me do that, because nobody wants to watch me do that. I'm going to be testing for 4 different biomolecules, and for each biomolecule I need 3 treatments. So now what I'm going to do is I'm going to take my test tubes. I'm going to have 4 sets of 3. Okay. This first set, what I'm going to do is I'm going to label SS for simple sugar. And I need a positive control. So I'm going to indicate that using a little plus sign here next to my SS. All right, this one's going to be a negative control. And this one is going to be my experimental group, so I'm going to write a E on that one. I don't know if you can see that. Okay. So now I have my simple sugar positive, my simple sugar negative, and my simple sugar experiment. Okay. Now I'm going to repeat that process. So that one's done. Oh wait, it looks the same. But everything's labeled now. Looks like my water's boiling, so I'm going to go ahead and turn that baby down to about medium. Now I'm just going to verify that I've arranged my tubes in their racks in a goof-proof way. As you guys will probably get sick of me talking about, I'm ADHD and I mess things up. So I'm going to make sure that I spend an extra moment to make sure that I don't screw this up. And that's a good thing to do whether you're ADHD or not. Okay, so what I'm checking to see is that I have all my simple sugars in the same row, all my starches in the same row, proteins in the same row, lipids in the same row. And then I'm going to check my columns. These are all, ooh, I'm glad I did that because these are not arranged in the same way. All right, it's time to start adding our solutions. Now, the negative control is going to be really easy because we are always going to use distilled water. Okay, and this water has been treated to remove all of the ions and the dissolved minerals and stuff that we typically find in our tap water. And so this is as close as we're going to get in this lab to having pure H2O. And so there's nothing else, allegedly, that is present in this than just water. So I'm going to make sure that I'm going to work with my negative controls, just again, double checking, making sure that I don't do a silly goober. And what I'm going to do is I'm going to add enough distilled water that it will fill up my tube until about that line. It doesn't have to be perfect. The great thing about biology is close enough is good enough. Now I'm going to do that for every single tube of my negative controls. The reason that we need negative controls when we're doing qualitative tests like this is so that we can compare our experimental group, right, our foods that we're testing, to something that we know is a negative result. Now, if we end up getting a positive result in our negative control, we know that something went wrong. There was some contamination somewhere. Somebody messed up. Might have been you, but let's blame the prep team because that's what I like to do. Okay. Or the data. Blaming the data is a good one too. Okay. So anyway, if we get a positive result in the negative control, we know that we need to try that negative control again, maybe run the whole entire test again so that we're making sure that our results are valid. The positive controls, however, are a little bit different, right? Because we are going to use a different indicator for each of the biomolecules. So for our positive control, when we are looking for simple sugars, which is the first test that we're going to do, we're going to use a 2% glucose solution. And as you probably recall, glucose is a monosaccharide, right? It's a simple sugar. So this should give us a positive result. So what I'm going to do is I'm going to pull out for all of my different foods that I'm going to test, I'm grabbing, whoopsies, just the simple sugar positive controls. That's it. And I'm going to go ahead and fill these with my glucose solution. That one was a little much. It's fine. Up to that line that I drew earlier. Next is up is starch. The positive control that we're going to use for starch is a 2% starch solution. So what you can notice though is that a lot of that starch is settled out. So all I got to do is just give that baby a little mixy mix. Okay, and I'm ready to grab all of my starch positives. We are going to use muscle milk. Okay, this is just protein powder that you would get at, a health food store, the thing that the scoop that you put in your shake in the morning. The last biomolecule that we're going to test for then is our lipids. Okay, so grabbing all of the lipid positive controls. Our positive control for lipids is going to be vegetable oil. We know that this is all lipids, so this should give us a positive result. Vegetable oil smells like donuts. I don't know what that says about the vegetable oil or what it says about me. Our lovely prep team went ahead and blended up the foods that we're going to test for. So here's our lettuce. It looks real chunky. We've got some milk. We've got some tuna. This is why you're glad that you're in an online classes, because you don't have to smell this. I do. You're welcome. And then we've got some refried beans. Again, we're seeing that these things have settled out, so we're going to give them a little shaky shake before we use them. We're going to make sure that we're not cross-contaminating our droppers, so this lettuce one only goes in the lettuce solution. now we're ready to start doing our tests. So I'm going to start with the simple sugars because remember we have to heat that. So I'm going to add 1 dropper full of Benedict solution, and remember that this needs to be heated. I'm going to add 1 dropper full to each of the tubes that I'm going to test. For those simple sugars. I have to go get more stuff. So we have our Benedict solutions all in all of our simple sugar test tubes. We're ready to put it in the hot water. But before we do, there's some important safety considerations that we need to make. I need to put on these super rad safety goggles so that if these tubes break when I put them in the hot water, I don't get broken glass in my eyes. If while this is sitting, if this liquid gets hot, it might shoot out of these test tubes, and I don't want that landing in my eyeballs either, so goggles. Okay, the other thing that we need to do to keep ourselves safe is use our test tube clamps, and this is going to set those carefully in there. Let me adjust the angle. Okay, we're just carefully setting these test tubes in the hot water. The Benedict solution needs to heat up so that it can interact with those simple sugars. And again, remember that we're looking for a color change from kind of this blue color, right, that we've seen in the initial color. And we're looking for changes to green, orange, yellow, red, somewhere along that spectrum. So timer. Now, while that's cooking, we can start doing our other tests. So let's start with the starch. We're going to record the initial colors of the solutions on table 2, which is your results table. It's on page 5 of your lab. It looks like that. Okay, and I'm going to have photos for you, so don't worry about not being able to see from way over here. We're going to start with our starch next. So we're going to be using grams iodine as the indicator solution. A positive result is going to cause the liquid to turn black or kind of sometimes a dark purple or a dark navy blue. But let's go ahead and give that a try. Okay, again, adding 1 dropper full of the solution to each of my starch tubes. Oops. So here's what our positive control looks like. It's pretty dark. Okay. All right, our next test is for proteins, and in this case, we're going to use the Biomolecules solution. If this is a positive result, we're going to see it go from this nice blue color to kind of a more purple. And just for reference, this is our positive control. Test is for the lipids. This test is a little bit different because the color change isn't quite so dramatic, and we have to let it sit for a little while, but we'll gather up all the results while we're waiting. And we're just going to give each of these tubes a little shaky shake. And then for contrast, this is the positive control. This is the negative control. So we definitely can see. Time to pull out the Benedicts. What we do, I'll show you the results.